We will continue now with our next speaker, who is Alice Kane from David Sinclair's group from Harvard. Hi, Alice.
Hi, thank you so much. Just get my screen going. Is that all fine? You can see?
Yeah.
Okay, great. Well, thank you so much, firstly, to the organizers for the opportunity to talk about our work today. I will be talking about age and life expectancy clocks based on machine learning analysis of mouse frailty. So currently, the discovery of novel interventions that increase health and lifespan in mice is limited by a lack of predictive measures of biological age and lifespan. So ideally, what we would have in a preclinical mouse experiment is that we would start an intervention at a certain age, perhaps 12 or 18 months, and then a certain amount of time later, perhaps 4 or 6 months, we would complete a standard assessment that would give us information about the health of the mice, but importantly, also give us predictive information about how long we expect these mice to live, so that we would be able to understand at this much earlier time point whether the mice are going to live longer and healthier, as shown by this green line, or hopefully not, live short, less healthy lives, as shown by this red line. Currently, there are some ways of measuring biological age in mice. And so the gold standard for understanding whether an intervention is delaying the aging process and increasing lifespan is to do a full lifespan curve. And as anyone who has completed one of these knows, these are incredibly time-consuming and incredibly expensive. So this is some important work out of Rafa DeCarbo's lab at the NIA, where they showed that metformin was able to increase lifespan. But as you can see, this study took more than 3 years and used 150 mice. Additionally, lifespan curves such as this, although giving us important information about how interventions affect lifespan itself, don't give us a lot of information about health. So we don't know whether the mice were also healthier as well as living longer. There's also been a lot of recent interest and a lot of talks in this conference talking about DNA methylation clock. So this is a model that's trained to predict chronological age on the specific methylation status of specific CpG sites. And these clocks provide exciting tools to measure biological age, but are unfortunately currently invasive, so require the collection of tissue. The analysis is also expensive, and at least in mice, they haven't yet been related to mortality. The third measure that I'll be talking about today is the frailty index. So this has come from a large clinical literature assessing frailty in humans, and is basically a clinical assessment of a mouse's health that's fast and non-invasive. And in fact, in humans, frailty indices are in fact the best predictor of mortality, outperforming other factors like specific disease status or even the DNA methylation clocks. So the mouse clinical frailty index was reverse translated from human frailty indices by Susan Howlett and her group at Dalhousie University. And it assesses 31 health-related deficits in a mouse. So this is just an example of one of these deficits. I hope you can see this video. This is a young mouse where we're assessing tail stiffening, and you can see that the tail is responding to the stimulus of the Sharpie well. Now we see an old mouse where there's basically no response of the tail to the Sharpie stimulus. So this old mouse would be considered to have this health-related deficit of tail stiffness. And there are 31 similar, simple, fast measurements that we complete in a mouse. So what we wanted to know was whether we could model these frailty measures in mice to predict both chronological age as well as lifespan. So to do this, we had 60 male C57 black 6 mice from the NIA. We completed the standard frailty assessment, the mouse clinical frailty index, every 6 weeks in these mice and tracked them to their natural death. So we then had this unique data set to be able to understand whether we could predict both age and lifespan. The approach we chose for this was a machine learning algorithm known as a random forest regression. And so this is basically a forest of 1,000 decision trees where data is input at the top here, in our case, our frailty data. Simultaneously, 1,000 decision trees determine the best prediction of our outcome, in this case, age, and we take the average of these to give our model. So we first used this approach to enter our frailty data and try and predict chronological age. And the outcome of this model we termed frailty inferred geriatric health timeline, or FRITE age. And we can see that this predicts chronological age in mice well. So we split our data into 50-50 training and testing. And if we just look at the testing side here, We have actual age of the mice in months on the y-axis, and we have their predicted age or fright age on the x-axis. And we see this beautiful positive correlation with a median error of less than 1.5 months. We next wanted to understand whether we could use these same frailty measures, but this time to predict remaining lifespan. So we used the same random forest approach, and this time we termed this model the analysis of frailty and death, or a frayed clock. And again, this nicely predicts actual survival in the mice. So here I'm just showing our testing data with actual survival on the y-axis and predicted survival or a frayed score on the x-axis. And we have a nice positive correlation with a median error of less than 2 months. So we were pretty excited about this because with these non-invasive, simple, fast assessments that basically any lab could implement, we're able to predict how long a mouse is going to live with an error of less than 2 months. Importantly, this also holds in mice of the same age. So if this clock just tells us that a 30-month-old mouse will die before a 21-month-old mouse, that's not particularly helpful. But if we break down this data into mice of the same age, so here each color represents a specific age group of the mice, we see positive correlations within mice of the same age between predicted and actual survival as well. Finally, we wanted to understand whether these clocks were responsive to interventions, given that our original aim was to accelerate discovery with these predictive measures. So to do this, we collaborated with Jay Mitchell and his group at the Harvard School of Public Health, and they had mice that were exposed to methionine restriction, which is a well-established dietary intervention that's known to increase health and lifespan in mice. And they had done a frailty assessment at 27 months of age, and so we were able to apply our models to that data. And excitingly, we saw that the methionine-restricted mice compared to controls had lower fright age scores, so lower predicted chronological ages, or perhaps lower biological age, and also had higher afraid scores. So the methionine-restricted mice were predicted to live longer than their control counterparts. And so we really hope that the aging field takes up the use of these clocks to accelerate discovery. And to this end, we have created a website, which you can see here. This contains a video with details of how to score frailty in the mice. And additionally, as you see from this short video, allows you to upload your frailty data, and the website will automatically calculate a fright age and a frayed score for each mouse and plot them if you would like. We're also very open to to collaborations to test whether other interventions are responsive to these clocks. So please reach out if you're interested in that. And I'd like to thank everyone in the lab, particularly Michael Schultz in the middle here, who co-led this study with me, as well as our PI, David Sinclair, our collaborators and funding. Thank you.
Thanks, Alice. We have some questions on Slack. So one from Morten. Great talk, Alice. I'm wondering which parameters are most important for the accuracy of your predictor?
Yep, that's a great question. So the random forest gives you this outcome of importance that gives you some kind of indication of how much the particular items are contributing to the models. Interestingly, it wasn't the same items that predicted age and lifespan, which I guess is not unexpected. So items that were particularly associated with lifespan were items that measured weight loss or weight change, as well as tail stiffening was a particularly predictive measure. I can't remember the others, but we have this, was actually recently accepted for publication in Nature Communications, so should come out any day and is available on bioRxiv as well. So please look up, we have the plot of all the importances.
Okay, great. Another question from Eva Van Gallen. Some interventions increase health span, but not lifespan. How would this program cope with that?
So, and data that I didn't show today, but in the paper, we also looked at enalapril treatment in collaboration with Susan Hallett's group, which is a treatment that's known to increase health span, but was tested in the ITP program and didn't increase lifespan. And our clocks were able to distinguish that where those mice had a lower fright age. So the enalapril treated mice were predicted to be healthier, but were not predicted to have a longer lifespan. So we do think that they can distinguish those separate outcomes.
Okay, thank you very much for joining us. And there's still a lot of other questions on Slack, so you can just go online to answer them. But thanks.
Great. Thank you so much.